Hello, my name is Roger Ramis. I'm a research professor at the IRB Barcelona, the Institute for Research in Biomedicine. And my today presentation is entitled Clinical Value of the MAF gain in Early Breast Cancer. These are my disclosures. Critical for breast cancer is the capacity of the cells to resist and relapse. We know that this is a process that occurs through a combination of facts. One is a probabilistic manner, but the second is also guided in a tissue and time-specific manner, particularly in breast cancer. This is, at the bottom, beautiful study published by Koenig and colleagues in Journal of Clinical Oncology in 2010. What you can see here in this multivariate analysis of site-specific and time-specific metastases is that ER-positive, hormone receptor-positive luminal breast cancer tend to metastasize first to the bone, and then they go to other tissues. And they do it in a low risk, but sustained in time. On the contrary, we have HER2-positive breast cancer. In this case, the likelihood to relapse occurs earlier in time, and then it flattens. And also what we can observe is an odds ratio for brain metastasis compared to other extraskeletal sites, suggesting that in this case, the treatment is pushing the disease to relapse in this specific tissue that is protected by the blood-brain barrier. In today's talk, we will focus on hormone receptor-positive breast cancer, as well as particularly in bone metastasis. In this cartoon, what we are trying to do is to depict how do we see the process of metastasis in the laboratory. In this case, what we are doing is simplifying this complex process into a multi-step process, in whereby a normal epithelial cell, in this case, the mammary breast, acquires a premalignant phenotype through the acquisition of mutations at the genetic and genomic rearrangement level. This leads to gain of oncogenes and loss of tumor suppressors. And in turn, we can also observe changes at the epigenomic and genomic instability that may also lead to these changes. All in all, this makes that a normal cell that obeys the rules between the body of behaving, growing to certain limits and exercise a given function, all of a sudden start disrespecting the boundaries and the limits where this cell can grow. The cells start expanding, they get immortalized, and eventually they transform through the accumulation of these aggressive phenotypes, and they generate what we define as a malignancy, which is the second main step. In this case here, what we have is a cancer cell that grows into a tumor mass. This tumor mass, in order to exist, needs to trigger self-renewal processes by these cancer cells. To avoid cytostasis, remember that the main signal that a epithelial cell receives is to stop proliferating at a given point. So the cells need to escape and avoid this signal, as well as to avoid or evade immunity. As this mass is growing, it will generate inflammation that it needs to be dealt with. Also, these cells will need to self-sustain the proliferation through autocrine and mitogenic stimuli. And finally, as the mass expands, building blocks will be necessary, so an angiogenic switch is required that actually facilitates the access to building blocks and nutrients. Similarly, a metabolic switch occurs, which makes the cells more inefficient in terms of managing resources, metabolic resources, but at the same time, they can grow in a much more accelerated manner. And finally, at the boundaries, we will start identifying some cells that trigger motility and migration in order precisely to escape from this primarily localized site. What we have learned also is that the action of a combined action of the oncologist and the surgeon that upon detection of this primary tumor, particularly in the breast, is resected. And unfortunately, we know that some cells will survive the physical and mechanical forces in circulation that will shred most of the disseminated cells apart. However, we know that some cells might overcome this either by triggering epithelial to mesenchymal transition process, as well as getting trapped in distant capillaries, in atheroma plaques, and eventually extravasating to a different sort of a distant organ. Importantly, here, some cells we have learned that will survive in what we define a dormant or a latent state. So cells that will not proliferate, so they will skip chemotherapy in the adjuvant setting. They just get arrested in G0, G1 until they receive a signal that triggers growth again and the generation of metastases. We also know that there's another type of dormant metastasis, which is those micrometastases where there is a balance between proliferation and death, likely triggered by the immune system. But as I pointed out, there's a point where these cells will get a signal to start triggering regrowth again, and this will generate a metastasis in a distant tissue as we truly understand it, which is through extravasation, homing and niching in this new environment, triggering mesenchymal to epithelial transition because metastases are epithelial, and then start cross-talking with the new environment and the new stroma cells. And they will trigger what we define as organ-specific colonizing factors. You can already anticipate that it's not the same for a cell, an epithelial cell of the mamma, to grow in the mammary fat pad, which is rich in adipocytes and therefore reservoirs of metabolites, than to grow in the lung where there are no adipocytes, but on the contrary, it's an area which is rich, or a tissue that is rich in oxygen. So this will encompass certain adaptations and modifications that are going to be extremely important. Now, we are focusing on bone metastasis, and in this schematic, what I'm trying to do is to summarize what is a normal bone or bone homeostasis, as you can see on your left side of the screen, versus what is occurring when there is a metastasis. What we know is that in a bone, in a normal individual, we have two main cell types. One is osteoblast and the other are the osteoclasts. One generates or synthesizes bone, that's the osteoblast, whereas the osteoclasts, which are large trophic macrophages, they degrade the bone. And it's the combination of the two activities that allows our bones to actually be a dynamic tissue, and a tissue that can heal upon fractures, and so that it's constantly adapting, for example, adapting to high exercises, thickening the bones, whereas adapting to more sedentary lifestyles by reducing the density of the bones. On the right side, what you have is what occurs when a metastasis is established in these bones. It usually generates what we describe, or is described in the literature, as the vicious cycle of bone metastasis. In this case, and you can see in red, what we have is that the cancer cells get into the bone, and what they do is at this stage, they trigger or they produce factors that tend to stimulate the differentiation of the pre-osteoblasts into osteoblasts. When this occurs, these osteoblasts start producing not only bone, but produce a lot of cytokines, including rank ligand. Rank ligand can be sequestered by OPG, but there's a point where this production of rank ligand overcomes the OPG, and then it signals through the rank receptor into the macrophages, particularly into the pre-osteoclast that will differentiate it into the osteoclast. Now, when these osteoclasts are differentiated, they start activating the degradation of the bone, and then what this causes is a massive degradation or liberation of growth factors into the environment that are sensed by the cancer cells and stimulate the proliferation of the cancer cells, thereby, again, fueling this sort of cycle again, triggering more osteoblast differentiation and osteoclast differentiation. Is the balance between the osteoblast differentiation and the osteoclast differentiation, which will lead to these bone metastatic lesions to become more osteoblastic, as in prostate in general, or more osteoclastic, as in breast, as can be seen in breast where it's largely dominated by the osteoclasts. Now, we know that these metastases in the bone are, associated with a lot of morbidity issues, such as pain, fractures, etc. And we also have learned that there are bone-modifying agents, which are agents that can manage this vicious cycle and interrupting it. Usually, we have two main classes of drugs. One is the bisphosphonates. These are drugs that either inhibit the action of osteoclasts or trigger apoptosis, such as clodronate or zoledronate. And we also have an anti-rankle antibody, denosumab, depending on which setting, called Xgeva or Prolia. Now, what we do know is that in all these situations, what it's produced is an inhibition of the differentiation activity or survival of these osteoclasts. And when you block this, what you block is this futile or vicious cycle, and therefore, bones are more resistant to fractures and not so much pain is observed. Now, what is important to point out is that despite the literature, now we have clear evidence that these type of drugs do not affect the cancer cells per se, and this is what is outlined in this slide. This is work by Peter Friedel and Eleonora Dondosola in Nijmegen. They did two-photon confocal microscopy of live activity of osteoclasts in bones in mice. On your left side, what you can see, particularly on the images on the top, what you can see is the action of some prostate cancer injected in in what is considered sort of a piece of bone. In this case, this is implanted in the back of the mice. This is an osteogenic niche, and once the cells are implanted, as you can see, with this hole made by a needle, the cells start generating and growing, and you can see how these cells start eating up the bone as the hole progresses and becomes bigger by day 9. If you now treat these mice with zoledronic, one of these bisphosphonates that blocks the activity of the osteoclast, what you can see is that now this degradation is stopped, so it no longer occurs. On your right side, what you can see is how the authors, again live, they monitor the activity of osteoclast, in this case by alkaline phosphatase, and what you can see is in the middle panel at the top is how in the controls there is an increased activity measured by the heat map of the osteoclast. If you treat with zoledronate, now this activity is reduced, as you can see by the reduction of the more reddish spots on the heat map. Now what it becomes very central, particularly as well for this topic, for the talk today, is the fact that if you measure the mitotic index or the number of mitoses of these cells in the bone, and you have this summarized on your right side at the bottom, what you can see is that irrespectively of whether these mice are treated with bisphosphonates and osteoclast activity is inhibited, there is no significant differences in the number of mitoses that are detected in these bones, which what this implies is that zoledronate or a biphosphonate actually acts on the osteoclast, but is absolutely uncoupled by the activity of tumor cell proliferation by the metastasis. Now, as you will see, this will become important later on. So we, in the laboratory, in the past, have identified an amplification, the 16q23, that is actually associated with an increased likelihood to develop bone metastasis in patients with breast cancer. On your left side, what you have is the amplification or the gain detected by FISH. As you can see, left top is control nuclei from a breast cancer patient. On the bottom, patient that has the gain or amplification. And as you can see, when we look at a validation cohort, in this case, and we look at time, specific time to bone metastasis, those patients that in the primary tumor biopsy have the amplification for the 16q23, they're at higher likelihood to develop a bone metastasis. And this is true for all breast cancer, as well as particularly for those that express the estrogen receptor. Importantly, this amplification predicts this likelihood of relapse irrespectively of any other parameter that has been detected in this patient, such as age, tumor type, size, etc. Now, what we were able to do back then, and this is published, is to identify who in this gain or amplification at the 16q23 is driving this effect, and this is a transcription factor of the AP1 family called MAF. And what we can confirm is that when MAF expression is high on these tumor biopsies on the primary tumor, then the likelihood of bone relapse is increased. B stands for bone metastasis. D stands for death, as death is used as a competing event in this analysis. Now, this can be summarized in the cartoon on the right. What we see is, again, an amplification or a gain at the 16q23 in the primary tumor. If this is there is more MAF, transcription factor, and this causes sort of a cascade of events that lead, and we could identify and validate, for example, to PTHrP accumulation. PTHrP is a cytokine that is extremely important at controlling calcium homeostasis, and it does so because it triggers differentiation of osteoclasts. So when these cells that have this amplification and produce PTHrP go to the bone, these trigger osteoclast differentiation and facilitate bone metastasis. Now, with this in mind, we went on to try to validate this into the clinical setting. Now, we started with the hypothesis that patients with MAF amplification may be more likely to benefit from the use of biphosphonates. And we were lucky enough to have access to both the AZURE and the NSABP-B34 clinical trials. Both trials have more than 3,300 patients enrolled. And in both trials, it was tested the intention to treat with a biphosphonate in the adjuvant setting to actually prevent bone metastasis. Importantly, both trials failed to meet their primary endpoint, and they show no benefit from the inclusion of the biphosphonate in the adjuvant setting in the overall population, albeit in some cases using subgroup analysis, some effect could be seen in some of the subgroups. Now, we had access to these tissue samples, and we use a sort of hypothesis generating and validation schema, first one of the trials, and then we move into the second one. And what we saw, and this was in collaboration with a company called Inbiomotion, what we saw in this setting was the following. To our surprise, the biology was fulfilled in the control arm. So tumors that have this gain and that express MAF are more likely to relapse into the bone. However, when we stratify those patients according to MAF expression and treatment, what we observed was, a bit surprising. What we observed is that the tumors that are not carrying the amplification or gain, which are also tumors at risk of bone metastasis, because in these trials, patients are enrolled with tumors more than 2 centimeters that are stage 1 to 3, and that they may, be at risk. So these patients that are 4 out of 5 that do not carry the MAF amplification when they are treated with the biphosphonate, what you see is that these patients improve in terms of IDFS, so they have less relapses. The significance is the differences are very significant and carried on time. And these actually translate, as you can see in the panels at the bottom, in an improved overall survival. And this is not a minor improvement in overall survival, it's quite significant in large. Now, we use the alpha in a hierarchical manner, so both, the two analyses were valid and were part of the primary endpoint. Very importantly, in these trials, what we use is we fulfill, the EMA 2019 guidelines, and we use we use a statistical analysis plan that was signed by all parties ahead of study start, and all the analyses were done blinded by an independent party. Now, what was very, reassuring is that the data that we obtained in the AZURE was actually confirmed when we tested the patients of the NSABP-B34, which was the confirmatory study. In this occasion, the biphosphonate was clodronate, not zoledronate, but again, we saw an improvement in disease-free survival for the non-amplified or non-gain patients that translated in an impact in overall survival. So the patients that were taking the drug were living longer than those that were not using it in the adjuvant setting. Now, how do we explain that we see a benefit in 80% of the patients, so 4 out of 5? Importantly, it's independent of any sort of known clinical pathological parameters, including menopausal status, ER, PR, HER2, even staging, size, etc. And when we interrogate 100% of the population, we don't see this difference. So the surprise came in the following slide. So when we consider and we look at the MAF positive patients, so MAF amplified or gain, what we observe, and here just to catch your eye, I'm focusing on patients that are non-postmenopausal, but this is the case for all the patients that are MAF amplified. What we observe is that the biphosphonates do not reduce the time to bone metastasis as first event, as you can see on the top panel on your right. But what it largely does is that if you take these patients that are very aggressive and you block the bone by using this biphosphonate that doesn't act on the tumor cells but on the stroma, what you have is that you massively displace the metastases to extraskeletal sites. As you can see here at the bottom, time to non-bone mets is massively increased, hazard ratio 6.6, and it's clearly significant. Look at the interval of confidence, and it's massively displaced by the action of zoledronate in this case. This is data from this room. So to sum up, patients that do not have the amplification to benefit from the treatment of or the inclusion in the adjuvant treatment of a biphosphonate, patients that do have MAF amplifications, if you include in the regimen a biphosphonate to try to prevent or to try even to manage, calcium-induced osteoporosis, because the doses that are approved in the guidelines are very similar, you are you may be incurring in potential harm to the patient, as it can be seen here in this data. These are all published, and you have here these citations. Now, how this compared to the current guidelines nowadays that are using, biphosphonates, or are recommending the use of biphosphonates in clinical guidelines. So nowadays, the patients that tend to be postmenopausal, they are recommended for the treatment with zoledronate. Apologies, here in your left panel, the percentage of menopausals are swapped. The majority are postmenopausal. For those, zoledronate is recommended. For the premenopausals, it's not. At the bottom, what you see is the meta-analysis published by the Oxford group, and how in the postmenopausal there seems to be a benefit, but it's minor, whereas in terms of absolute risk reduction, whereas the premenopausal, no effect is seen. However, if we apply the MAF test and a biomarker, what you can now see is that if the patients are MAF negative, and this is 80% of the patients, clearly there's a benefit, and this is of a larger magnitude in terms of absolute risk reduction, in terms of IDFS, and overall survival. But more importantly, if a patient is MAF positive, it's likely that this patient should not be treated because there is a likelihood to cause harm on it, as you can see, by negative absolute risk reduction in terms of IDFS, and also because this translates into a poorer overall survival. So that, would sum up the status and probably led us to a sort of opt-in, opt-out on the basis of the biomarker, and for the first time explains why those large trials, the Azure and the B34, actually did fail to provide, significance and the benefit. Remember, a subgroup of patients will benefit, which is very large, and the other subgroup of patients that is MAF positive, they should not be treated because it's not that the drug is innocuous on these patients, but it may cause some harm. Anyway, with that in mind, and also that's what I was mentioning before, the doses that are recommended for biphosphonates in the adjuvant setting are exactly the same that are recommended for cancer-induced osteoporosis. So, this actually raises the concern sometimes that maybe when treating for the cancer-induced osteoporosis, this may, again, may cause some harm, potentially some harm in the MAF-positive breast cancer patient. Anyway, so we decided to try to investigate how these, MAF-positive patients are so prone to metastasize elsewhere, and to molecularly explain how this occurs. And we're very happy because this story was selected as one of the highlights for the San Antonio Breast Cancer Conference on 2023. Anyway, what we did in the laboratory was first generate a mouse model that recapitulates what is occurring on the patients. So that means that we introduce in the ROS26 locus the MAF transgene that is controlled with a stop promoter in front of it that it's LOXP flank. So that means that upon the induction of a Cre recombinase, this can be excised, and then these cells will express MAF. And this is indeed what happens. We have these mice. These mice do not express MAF. We induce tumors in these mice with progesterone and DMPA. These tumors are ER positive, as you can see here described on the top panels. ER positive, they express the cytokeratins, etc. And then what we do is we put them in culture, and we take the cells, we divide it in two pools. So we have cells that are isogenic. They mean they have the exact same mutational landscape. And then in one of the two sort of populations, we activate MAF. And what you can see at the bottom panels is that indeed the activation of MAF, it's very exquisite, nuclear staining, and we can monitor. Now we can compare how the cells with the exact same mutational landscape to behave in the presence or absence of MAF in terms of bone metastasis. And what we observe is the following. Is if we inject these cells intracardiacally into the left ventricle and we disseminate them all over the mouse, what you can see in yellow is that the cells that, and by the way, this is in the syngeneic background, so that means that the mice have immune system. So if you inject these cells into one of those mice, the mice will barely develop metastasis, and if they develop this metastasis, will be very small, as you can see in yellow in the top panels. Whereas in blue, you have the cells with the MAF, and in this case, the cells go into the bone, they survive better, and eventually they take up and they generate very strong and heavy metastatic lesions, as you can see at the top right images. Now, the other thing that we tried to do is to recapitulate what we're seeing in the patients, and then we explore what happens in terms of bone metastasis. We treat with zoledronate. Bottom left, what you have is the incidence of bone metastasis versus non-bone in these mice that have MAF amplification whether they are treated with PBS or zoledronate. And clearly there are no significant differences, and if anything, it seems to be a higher trend towards bone metastasis. Now, what was quite impactful is what happens on the right side on the panel on the right at the bottom, the number of extraskeletal metastases. And in this occasion, what you can see is the treatment with zoledronate. What it does is that massively increases the number of extraskeletal metastases in those mice, again mimicking exactly what we described already in the clinical trials. So that it was very important from our end. So we have a mouse model that exquisitely recapitulates exactly what we are seeing in those patients that have a MAF amplification and that are treated with a biphosphonate. At the same time, we use a sort of a bottom-up approach. We decided to interrogate breast cancer cells, human, that are hormone receptor positive, so that express ER, to interrogate them, what is binding or interaction with the MAF transcription factor. In this occasion, what we did is we take the MAF transcription factor and we express it in the cells at very low levels, but together, either in front of the MAF protein or at the end of the MAF protein, at the end or the C-terminal of a small biotin ligase, BIR-A. Now, what you can do in those cells that express this MAF at very low levels, but with this catalytic enzyme at one end or the other, is that you can do a pulse chase of biotin, and then MAF, and particularly the biotin ligase coupled to MAF, will paint around all the proteins that are in close proximity to MAF. Then what we can do is we can lysate the cells and pull down and do mass spectrometry to identify which proteins have been painted by MAF. In other words, which proteins are in close proximity to MAF. And we look at that with the two different MAF isoforms at one side or the other of the protein, and we'll look for what was common. And this is summarized on this distinct plot here, if you follow the arrow on your bottom left. Now, what caught our attention, and this is summarized as well as in the right side, what caught our attention, because it was an interactor, was very specific for hormone receptor positive cells, is that MAF interacts with estrogen receptor itself, which all of a sudden to us was, wow, this might be very important because we know estrogen receptor is central for the treatment of hormone receptor breast cancer, and these represent a large majority of the breast cancer. So with this in mind, we went on to evaluate how relevant was this interaction between MAF and ER in terms of phenotype, in terms of functions that are provided to these cells. And what we saw is that it facilitates the tumor-initiating cells. It also enhances or has a synergistic effect in terms of proliferation of the cells, as you can see as BRDU incorporation and nucleotide incorporation into DNA. If you have MAF plus estrogen, this is a lot faster than if you only have estrogen. And MAF also itself facilitate migration of the cells, adhesion specifically to bone marrow derived cells, and media produced by MAF expressing cells. It's a lot better to actually differentiate osteoclasts in vitro that the same media produced by cells that do not overexpress MAF. So all in all, MAF and its potential interaction to ER was being very specific for certain functions that are needed in order to metastasize, and particularly to the bone. So we next evaluate in terms of gene signature or gene expression, what changes are we identifying in these cells in the presence of MAF and estrogen. So for this, we take these human cells, MCF7, hormone receptor positive, with and without MAF, and we treated them with hormone-deprived media or with estrogen. We then did RNA-seq, and this is the heat map that you have here in the middle. Cluster 1 and cluster 2 actually reflect on the genes that are specifically induced by MAF or downregulated by MAF, irrespectively of estrogen. Cluster 3 and cluster 4 account for the usual response to estrogen of hormone receptor positive cells. And cluster 5 and 6 caught our attention as being the most relevant for us, because these are two specific clusters that were previously unappreciated that actually increase its expression in the presence of MAF and synergistically in the presence of estrogen and MAF. In other words, the combination of the two stimuli, estrogen binding to estrogen receptor and MAF, has or generates this previously unappreciated gene transcriptional program that may support metastasis. And indeed, if we do a close lookup at them, what we identify is that in these clusters, particularly in the genes that are upregulated, we contain several estrogen early and late responses, activation of inflammatory responses, and TNF-alpha, as well as epithelial to mesenchymal transition genes. Some genes that contribute to this or that are part of this is FGF18, which is a fibroblast growth factor known to contribute to migration and EMT, PTHLH, also known as PTHRP, as I already pointed out, known to be a MAF target that is critical in bone formation and bone remodeling. We also identify JAK1, promoter of bone metastasis and activating Notch signaling, as well as TGF-alpha, JAK1, and Sonic Hedgehog. We can validate this by PCR, and we can also validate this in the tumor genome cancer atlas, as we can see, or in the metabolic cohorts, as we can see, the expression of these genes do actually correlate with the expression of MAF in these tumors, in breast cancer tumors. Anyway, with this in mind, we move on and we try to understand how come that MAF and ER with estrogens are interacting and facilitating this transcription. And we look at what happens in terms of binding to the chromatin. This may sound a little bit complex, and we'll try to summarize with what is more important. Just focus on the Venn diagram in the middle. This Venn diagram, what it does is it provides you, in the absence of estrogen, it's what's called MOC, hormone deprived. It provides you the number of sites where the estrogen receptor binds in the genome, which is 595. Now, if you add estrogen, and estrogen, remember, binds to estrogen receptor, and this goes into the nuclei, and it triggers genes such as MYC, cyclin D1, GREB2, etc. Now, the estrogen receptor binds to up to 2,837 new sites, in addition to the previous ones that are bound. So clearly, estrogens facilitate the function of estrogen receptor, as it's well known, and that's why you treat with tamoxifen and some estrogen inhibitors. Now, the critical question is what happens in the presence of MAF? And so here we go. So if you have the same cells in the presence of estrogen and MAF, now instead of binding to these roughly 3,000 sites that estrogen is facilitating, now this estrogen receptor will bind to 5,000 sites. So more than 2,000 extra sites in the genome will be bound by estrogen receptor. And now you can already start to calibrate the magnitude of the effect of having MAF amplification. Just considering estrogen receptor, and you know how important is estrogen receptor for breast cancer, in the presence of MAF, estrogen receptor is now licensed to bind to many more sites in the genome. And the consequences of this extra license is that now estrogen receptor can kill and can drive bone metastasis or metastasis in general. So how is MAF facilitating this? So we went back into our interactome and look at who was actually binding to MAF. And we came across KDM1A. What is KDM1A? KDM1 is a lysine demethylase that is very important to rearrange the chromatin, and particularly to facilitate closure and opening of the chromatin. So why do we focus on KDM1A? Well, we have seen that MAF is amplified in breast cancer, and this leads to bone metastasis. We also know that MAF is amplified or overexpressed in multiple myeloma and angioimmunoblastic T-cell lymphoma. Both the type of non-solid tumors do also metastasize the bone. Now, interestingly, in the literature, it has been reported that KDM1, also known as LSD1, is also important in multiple myeloma to do metastasis into the bone. So we felt very compelled to see that these two could interact, again, not in multiple myeloma, but in the breast cancer, and particularly, again, to do bone metastasis. So we explore how relevant was KDM1. As I said, lysine-specific demethylase. This is very important. And we decided to explore how many of the genes that were targets of MAF and estrogen were dependent on KDM1. So we induced a genetic depletion of KDM1, and we did RNA-seq to look at what genes go up and down in the presence of MAF and estrogen, and how many of those are dependent on KDM1 absence or genetic depletion. And as you can see, a substantial amount of the genes that we were interested in are dependent on KDM1, including SOX9, JAK1, PTHLH, FGF18, which we validate by quantitative PCR in bulk. Anyway, what is important though is that now with this in mind, we can go back into our model and have a proof of concept of how relevant is KDM1 to facilitate MAF colonization of the bone. Here we can use ORI1001, which is an inhibitor specific for LSD1 or KDM1 that is in clinical development in phase 2 for multiple myeloma. This inhibitor blocks KDM1, and when this is blocked, there is an accumulation of lysine to histone 3 dimethyl. That means we're inhibiting the dimethylase function, and methyl groups are being accumulated. Now, what we can do is induce the dissemination of the control and MAF expressing breast cancer cells into the mice, and then we monitor bone metastases. And you have the summary of the results on your right side. In yellow, what you have is the control group without treatment. So what you can see is that in the majority of the cases, there are very few, there are very small metastases or no metastases at all, as we already reported earlier. If you treat with ORI1001, there are barely metastases, again, very small metastases, minor metastases. That's in red. In blue, what you have is what happens when you put MAF expressing cells. You have many more metastases and larger metastases. Remember, the scale is a log scale. So what you have is, many more and bigger metastases. And then in green, what you have is the effect of treating these mice with ORI1001, which is this KDM1 inhibitor, which actually will block the capacity of the chromatin to be open in the presence of MAF. And when we do that, what you can see is that we reduce the number of metastases dramatically. Actually, we cure the vast majority of the mice. So these metastases cannot grow into the mice. And the reason is because although they have MAF, they cannot open the chromatin to facilitate the function of MAF, increasing the number of sites where the estrogen receptor can bind. And this is summarized in this cartoon. What we see is that estrogen receptor usually binds to a series of enhancers in a regular breast cancer cell. If MAF is there, MAF licenses these breast cancer cells to actually, and estrogen receptor to actually drive breast cancer metastases. And it does so because it interacts with the chromatin remodeler that facilitates opening of the chromatin in previously unappreciated sites where now estrogen receptor can bind and can trigger metastasis. And again, just to wrap up, and as a summary, and after I hope this 20 minutes of presentation, I hope that I have convinced you that we have mapped how MAF amplification or gain licenses breast cancer metastasis cell fate. Indeed, the amplification of MAF and its expression is critical, or it's a big plus to actually facilitate breast cancer bone metastasis and metastasis in general. MAF expression facilitates this process because it leads to a global chromatin opening at the enhancers, and it does so because it interacts with the chromatin remodeler, KDM1A, and when it does this, it facilitates the interaction of estrogen receptor to previously unappreciated promoters and the transcription of genes that are extremely important to facilitate this process of metastasis. And it's not one gene, it's many genes, and that explains why one gain has this downstream so expansive phenotype. More importantly, I think from the clinical perspective is that MAF amplification enables a precise use of biphosphonate in the adjuvant treatment. We can now deliver, beneficial biphosphonate treatment to those patients that are negative for the MAF amplification, and it's, avoid unnecessary treatments for those that are MAF positive that either will not respond or will be harmed by the therapy. And this is irrespectively of any other clinical pathological parameter that has been used in the past. And more importantly, also, we can explain finally why those large clinical trials have failed in the past. And with that, I thank you for your attention, and I'm looking forward to see this in the clinical practice.